2013, there were specimens of both Ipomopsis aggregata and Ipomopsis ramosa that were collected from the Roaring Forks Canyon. And then in the fall of 2013, Ross taught a systematics of botany class, and I was in it, and we got to sequence some genetic regions of these species. We did one chloroplast and one nuclear, and then we combined these with a recent large survey of phylogenetic just phylogenetics, and we examined it specifically in the subgenre of Ipomopsis. And this is our chloroplast gene region data, and what's most important to see here is that Ipomopsis aggregata, subspecies aggregata, and Ipomopsis ramosa share almost the exact same chloroplast genes. And so this can only be inherited from the maternal lineage or the seed donor. And what this points to is that because this is found on the Roaring Forks site, that Ipomopsis ramosa actually speciated on the Roaring Forks site, and that Ipomopsis aggregata, subspecies aggregata, is the maternal donor. And here we have our nuclear gene data, and we examine this because it gives us insight into both the maternal and the paternal lineage. And when we look down here, we see that Ipomopsis ramosa is in the same clade as Ipomopsis polyantha and Ipomopsis sancti spiritus, which are also narrow endemics found in this region. And so even though these are narrow endemics in this region, the likelihood that they speciated with Ipomopsis aggregata is highly unlikely due to the endemicity of both of these species. And with the data that we have, we actually don't have enough to see where the genetic donor is, but we do know that it came from an outside area. And so this leads to our hypothesized origin of Ipomopsis ramosa. So suppose that a long-distance pollinator was carrying foreign pollen and it found Ipomopsis aggregata and formed a hybrid. Could this hybridization have led to changes in floral morphology and in turn also led to a shift in pollinators, which led to its reproductive isolation? You want to tell us more about this, Meredith?
Yes, I will elaborate. So yes, let's talk a little bit more about this. Okay, this brings us back to the Papilio rutilis and the western tiger swallowtail. When we examine the last slide, as Shilah stated, these guys are pretty narrow. They're narrow endemic, so the probability of them hybridizing in Roaring Forks Canyon with Ipomopsis aggregata, subspecies aggregata, is highly unlikely. And so for that reason, it probably, the pollen candidate that led to the hybridization of the first speciation and the first specimen of Ipomopsis ramosa probably didn't come from this area. And so that's where we look back at our pollinator here. Keep in mind that we only saw it one time. And so what does that mean? Well, I'm thinking, what we're theorizing is that this tiger swallowtail probably brought in the first pollen donor, and it came from an unknown area, somewhere outside of the Four Corners region. And it just happened to leave this pollen in Ipomopsis aggregata, subspecies aggregata, and Roaring Forks Canyon, and the first specimen came about. But this is where our Hyles lineata comes into play. The tiger swallowtail only visited the canyon very briefly, but the Hyles lineata was there for an extended period of time, much longer than the tiger swallowtail. So we're theorizing that if the vector pollinator, the western tiger swallowtail, initially brought the first pollen grain, then it's possible that it resulted in the hybridization event and that Hyles lineata sustained the population and continued its reproductive success. So that being said, like most biological studies and any research, we answered some of our questions, but it only gives rise to more. And as far as that is is concerned, I think that some of the research that could possibly be looked into with maybe other senior thesis students that want to continue with this research is an extended phenological study. I did say that Ipomopsis ramosa has a 2-year reproductive cycle, but as a senior thesis student, I can only look at 1 year's worth of data. So it would be interesting to continue the phenological study in that I would be interested to see if the precipitation patterns still correlate. So say we had a drier year, would we see that spike in rosette later on in the season if there wasn't as much water availability? And another thing that I kind of left out, we didn't really talk about, was nectar-sugar contact and refractometry. We attempted to do this, and what it is we took a refractometer, which you add a little bit of nectar from your sample, your species sample, and you look at it in the light. And what it can do is it can tell you the amount of sugar in the nectar. The interesting thing here is that we were unable to extract any nectar from either the aggregata or the ramosa. And this is interesting because both of the pollinators that we observed in the field are generalists and tend to favor species that not only prefer plants with lots of nectar availability, but also with high sugar content. So what's the relationship there? If the nectar isn't necessarily the reward, what is? And so that leads into continued pollinator observations. If we hadn't been in the field that day to see the tiger swallowtail go through the canyon and pollinate Ipomopsis ramosa and catch it, we might have missed it completely. So it's possible that there are other pollinators that we haven't even seen that have specific migratory patterns that are very short-lived and also contribute to the speciation. And then one other thing I want to point out is Ipomopsis aggregata, as stated earlier, is not. It can exist in lots of different kinds of soil. However, the Ipomopsis ramosa is really specific. So what's the relationship between the Permian Cutler Formation and Ipomopsis ramosa versus its maternal donor, Ipomopsis aggregata, who can exist in all types of soil mediums? And lastly, further genetic research into the nuclear gene region. We looked at only one nuclear gene region, but there are many. And so it would be interesting to continue that study because we would really like to figure out where the parental lineage comes from, and probably from another area. So with that being said, I just want to thank everybody for coming out. And I want to thank you guys because this was so much fun. I mean, if we weren't out in the field with black lights trying to find pollen analog dye, we were chasing insects around in the field, or we were bushwhacking trying to find new populations, and it was a lot of fun. And so just to spend your summer in the field doing research, I mean, you couldn't have asked for a better thesis. And also to Al for letting us examine your baby and continue the research on the ramosa. It was a lot of fun. Thank you for finding it. Right? For finding it. And
It was all a setup. Ross and I decided to do it so you guys could have something to do.
well, we thank you for that. To the Native Plant Society for backing us and for funding us, and to Fort Lewis College, specifically the Department of Biology, and also the Botanical Society of America. They backed us on this one as well. And then last but not least, you guys know this guy, right? Okay. Human development here, don't you agree? This is our knight in shining green Subaru Forester, and he was there when we needed him. When we had questions, we could come to him, and he could help us make sense of just the amount of data we had. We had so much data, and to just bring it all around and to just make sense of everything that we got in the field, like we're really grateful. So thank you. With that being said, I want to open it up to question and answers, and we'll try and answer them to the best of your ability. Our ability. Yes. Do you want a microphone?
No, did you find any white Ipomopsis in the range of the planting?
When we observed the pollinator in action,
Is there any
specifically?
up further to the northwest? Are there any Ipomopsis that are white that could cause cross-pollination with the aggregata? Otherwise in Idaho or east of Washington.
I don't know the extent of all the Ipomopsis species because there are many. This tends to be a plant that hybridizes pretty easily. I will say that in the Roaring Forks Canyon, and not necessarily within our plots, but on Site H hillside, I did see some white Ipomopsis ramosa. but they were ramosa. They just tended to have less of that salmon
Now,
pigment.
I'm way north, like Idaho. Is there anything that could have been brought down to cause the
We
original?
didn't actually look at species that were in Idaho. When we looked at that subgenre of Ipomopsis
It was
in that
localized.
section, we looked at mostly things that are found in the Four Corners region.
Yeah.
And part of that has to do with the extent of the pollinators that we found. So we specifically looked at the Ipomopsis in that region because of the swallowtail butterfly. If you look at its migratory patterns, it only moves up from the
Tends to
southwest.
move north, yeah.
So with the pollinator evidence that we found, we don't think that there would be any movement from those northern species. Now, maybe if we were to continue to do observations and see potentially other pollinators moving around, we might see something else.
And did you find any beetles? When I take photographs, often I find a beetle in the middle of it.
You know, there was one really tiny, like pin-sized little black beetle that I saw. But it wasn't, I didn't see it all the time. I think I saw it only once or twice. And I attempted to put it in the jar, and the second I put it in the specimen jar, it disappeared. That's how little it was. So it's possible, it's highly likely that there are other pollinator specimens, but these are the two that we found.
You mentioned the Botanical Society of America. Are you going to publish in it?
That would be nice. That would really be great, put this on the map there, so.
They're very open to this.
Yeah, they were pretty excited about our project, so, yeah. I'm tempted, we did film this, so I'm tempted to maybe send them like a YouTube link. Question? Yeah? Yep.
Did you determine the method of seed dispersal, and do you think that it's possible that there were several hybridization events that caused these isolated populations?
No, I didn't quite get that far into it. I was mostly trying to figure out we didn't know how it reproduced to begin with, so I was trying to catch the fruits before they disperse their seeds, just to see that different factor on types.
This is the interesting thing about this research is that because it's so newly described, there isn't much information on it. And so we really had to start at ground zero, gathering the information that we could. And that's what I was saying about future research. If any of you guys are going into the biology field and are looking at doing this kind of stuff for research, you can take this and run with it. We've got baseline data for you. There's so many different directions you could go, including, let's see, she suggested. Yeah, absolutely.
Where did you get your precipitation data? Did you actually have rain gauges out there on the site?
No, we didn't. That was the national, help me out here.
That was a USGS station that's about 6 kilometers north.
Especially during monsoon season, it's very spotty, you get as far as what rain. I just wondered if it would be easy to stick a rain gauge right along the plot sites.
Yeah. Unfortunately, we didn't think a lot about that until later on, and it's a little bit late. But also, how do you document melting of snow? I mean, and that was a big part of May.
You
So.
gotta get some soil moisture.
Right,
But I'm
absolutely.
just curious, you described, you correlated the formation of the rosette and then you bolted, you kind of looked at the climatic cycle. Did you notice, and you may not have had time to do this, did you notice any differences in that and aggregata? Was aggregata behaving differently? Did they both tend to form rosettes and bolt at the same time?
Well, like we said earlier, where I did my phenology study was at site H, and this was mainly Ipomopsis ramosa. So there were hardly any Ipomopsis aggregata, only where Melanie was doing the study, where she directly put them next to each other. So a lot of the times where my plots were consisted mainly of Ipomopsis ramosa, and that's what I was documenting. I will say that the rosette stage looks very different. In the Ipomopsis ramosa, you get all these trichomes and it's super furry, whereas the Ipomopsis aggregata is more bare. I did not document the phenology of the aggregata. However, I bet you could find that information online because it has been studied pretty extensively.
Just curious how different.
Yeah.
So you think the parent hybridization is kind of following up on Bob was talking about the ability to come from the south somewhere?
Yeah.
So,
Mhm.
so something down in New Mexico is the parent, and a butterfly presumably picked that up and brought it up here and caused the hybridization.
Right.
And the moth is perpetuating
And that's where you see the switch and the pollinator switch. Yeah. Because that one stuck around and was able to keep the population going, whereas we think with the tiger swallowtail, it was probably only one event. And it's possible, I mean, I don't know the specifics, but it's possible that with a lot of the other narrow endemic species that you see in this area, that something similar could have happened.
That's what I was gonna ask,
Yeah.
have you compared it, any data, any research? You mentioned Sanctus spiritus and the one at Pagosa, Polyanthus. Have there been similar studies that you could compare?
I know we actually have another student here at Fort Lewis that studied the polyantha, I believe, in depth. And she might know a little bit more about the pollinator, but no, I have not yet compared her studies to mine. She did just do this year as well.
Anyone's done the pollination work with some experience for that.
There's so many other
Yep.
questions that arise.
Oh,
I mean, that's the beauty of biology, and one of the things that I like about it is evolving. So, is anybody else, anybody in the back? Students? Did we make sense?
Just another quick question. Did you see the moth also visiting Aggregata?
We did.
So it's visiting both kinds, but because of the position of the anthers, where the pollen is landing on the blossoms, that's what's potentially the difference. It's not cross-pollinated just because of different tubes.
So ramosa could go to aggregata, but not really the other way around, based on the floral structure.
And for other students who are interested in this, remember that one Sanctus spiritus is right outside of Santa Fe. So you get to spend a lot of time in Santa Fe.